Usually when we talk about a medical diagnosis, there's this expectation of precision, like engineering almost. You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is.
Right, it's binary, like it's broken or it's not broken.
Exactly, but then you step into the world of advanced oncology and cellular therapies, and suddenly that x-ray machine feels completely inadequate.
Yeah, totally.
We're looking at a therapeutic landscape that is just incredibly complex. So, welcome to the deep dive. Today, we are taking a stack of research on the cutting-edge world of RNK cell therapy in solid tumors, and we're going to deconstruct the exact mechanisms at play here, just for you.
And it's a landscape that is moving incredibly fast, almost daily. But to understand why this new therapy is causing such a stir, we really have to look at the baseline. Like, where are we coming from, right?
Right. So I want you to imagine a revolutionary cancer treatment that works absolute miracles. Like, it can cure previously incurable blood cancers.
Which is amazing.
It is. But there is a massive catch. It's staggeringly expensive. It takes weeks to custom build for each individual patient, and it can trigger deadly side effects. That's the current reality of CAR T cell therapy.
Yeah, that's the perfect way to frame it, actually. CAR T relies on what we call autologous manufacturing.
Which means it comes from the patient, right?
Exactly. We have to extract the patient's own T cells, engineer them in a lab to fight the cancer, and then put them back. It's a highly bespoke process.
And the side effects you mentioned, they are no joke.
Yeah, tell me about it.
We see cytokine release syndrome, or CRS, which is essentially a massive systemic inflammatory storm that can literally shut down organs.
And in the source material we're looking at today, there were some really surprising neurotoxic effects mentioned too, like non-classical Parkinsonism. What is actually happening there?
It's alarming, isn't it? We're seeing movement disorders emerging after treatment, which strongly suggests the engineered T cells are either physically crossing into the brain's basal ganglia, or they're triggering just profound neuroinflammation.
Yeah, it just highlights the desperate need for a safer, more accessible alternative to T cells.
Which brings us to the actual hero of our research today, natural killer cells, or NK cells. I understand they're part of the immune system, obviously, but why are they suddenly the superior platform for an off-the-shelf treatment?
It really comes down to a fundamental biological difference in how they recognize threats. So T cells are part of the adaptive immune system. They require strict matching of human leukocyte antigens HLA types.
The tissue matching stuff.
Exactly. If you put donor T cells into a patient without matching those HLA types perfectly, those donor cells will look around, realize they are in a foreign body, and attack the patient's healthy tissues.
Which is graft-versus-host disease, right?
Precisely. It's a fatal condition.
So that's why CAR T has to be custom built from the patient's own body. You can't just share T cells.
Exactly. But NK cells, they are part of the innate immune system. They do not cause graft-versus-host disease.
Meaning, you could theoretically take healthy donor NK cells, engineer them, and just give them to anyone, like a universal blood donor, but for cancer-killing cells.
That's the dream. That's exactly it. But how they naturally kill cancer is what makes them truly remarkable platforms to build on. They have dual innate killing mechanisms that T cells just don't have.
Okay, let's unpack this, because the details here are wild. First is something called missing self-recognition, right?
Yeah, missing cells. So think of it like a molecular ID badge. Healthy cells display a specific molecule on their surface called MHC class I.
MHC class I. Got it.
Right. NK cells constantly patrol the body, checking these badges. If an NK cell bumps into a healthy cell, it sees the MHC class I badge, gets an inhibitory signal, and just moves on.
It says, cool, you belong here.
Exactly. But tumors are sneaky. To evade T cells, which use those badges to find targets, tumors often hide or stop producing that ID badge entirely.
So the tumor makes itself invisible to the T cells, but the NK cell realizes the badge is gone.
Yes. The NK cell detects the absence of that inhibitory signal. It realizes, hey, this is a rogue cell, and immediately triggers apoptosis. It essentially forces the tumor cell to self-destruct.
That is brilliant. And there's a second mechanism too, right?
Yeah, antibody-dependent cellular cytotoxicity, or ADCC.
Okay, a bit of a mouthful.
It is, yeah. But essentially, if a doctor gives a patient antibody drugs that stick to the outside of cancer cells, the NK cell has natural receptors that recognize the tail end of those antibodies.
So it uses the drug as a targeting laser.
Pretty much. It sees the tag, locks on, and releases lethal toxins.
Okay, so if I'm visualizing this for listeners, if CAR-RT is a bespoke, hyper-expensive sports car that has to be custom built for one driver, and sometimes the engine overheats and causes a catastrophic fire.
Meaning the cytokine release syndrome.
Right, the CRS. If that's CAR-RT, then CAR-NK is basically a mass-produced, heavily armored tank. It's universally compatible, it's safer, and it's ready to deploy instantly.
That is a highly accurate analogy, honestly. But the catch is, if we want to deploy an army of these universally compatible tanks, we have to figure out how to build them at an industrial scale.
Right.
And that is not easy.
Yeah, because if we're not taking them from the patient, where are we getting them? Our sources break down 4 distinct cell sources for manufacturing these therapies, and they all seem to have pretty significant trade-offs. Let's start with the most obvious one, just getting them from regular donated blood.
Peripheral blood, yeah. It is easy to access. I mean, you just draw blood from a healthy donor.
Right.
But the natural frequency of NK cells in peripheral blood is very, very low.
So you don't get enough right off the bat.
Right. You have to aggressively multiply them in the lab to reach a dose large enough to actually treat a tumor. And this prolonged activation causes the cells to suffer from exhaustion before they even reach the patient.
They're tired before the fight even starts.
Exactly. Plus, you have high donor variability, like one donor's immune cells might be robust, while another's might be sluggish.
So if a standard blood draw is too variable and the cells get tired, what about umbilical cord blood? Because that comes up a lot.
Now, this is where it gets really interesting. Cord blood is a powerhouse. It's highly proliferative. A single cord can generate enough clinical-grade KRRNK cells to treat multiple patients.
Wow. And what's fascinating in the research is this very specific algorithmic predictor for success that emerged from recent clinical trials. I loved this detail in the notes. They essentially found a mathematical formula for the perfect donor.
They did, yeah. Researchers found that not all cord blood is created equal. They need cord blood units that contain fewer than 80 million nucleated red blood cells.
Fewer than 80 million.
Yes. And the time from collection to freezing has to be 24 hours or less.
Yeah.
If they meet those strict criteria, the clinical outcomes are vastly superior.
And if they don't, what happens if, I don't know, it takes 36 hours to freeze the cord blood?
It's crazy, but the resulting NK cells actually upregulate internal genetic programs for inflammation, hypoxia, and cellular stress. Basically, they're born exhausted.
Yeah.
They will not fight the tumor effectively.
It's wild how fragile the starting material is. Okay, the third source is pluripotent stem cells. So, iPSCs. Stem cells always sound like the holy grail in medicine.
They offer a true blank slate, which is great. You can genetically program them at the stem cell stage, which is much easier, and they offer unlimited expansion. You could theoretically make an infinite number of doses from one single source.
But I'm guessing there's a downside. There's always a downside.
A significant one, yeah. Coaxing a stem cell to develop into a fully mature, lethal NK cell is incredibly time-consuming, and often they get stuck in an immature state.
What does that mean, practically?
It means they have low expression of crucial activating receptors like KIR and CD16, which they need to kill cancer. And because you are heavily reprogramming their genetics, there's always a lingering risk that they can undergo malignant transformation themselves.
It could become cancer.
Yes, exactly.
Okay, so we have adult blood, cord blood, stem cells. And then there's the fourth source, the NK92 cell line. The backstory here is genuinely wild. The research says these cells were derived from a 50-year-old male with lymphoma all the way back in 1992.
That's right. It's what we call an immortalized cell line. They just keep dividing forever. They're incredibly easy to grow to a massive therapeutic scale. And because they all come from the exact same original source, there's almost zero variability.
Wait, let me pause you there. If they were derived from a patient with lymphoma, aren't those cancer cells? We're injecting cancer cells to cure cancer?
You've hit on the exact danger. They pose a severe tumorigenic risk. So to prevent them from forming secondary tumors in the patient, every single NK92 cell must be irradiated with radiation before it is infused.
But doesn't irradiating them basically give them a death sentence?
It does. It severely limits their lifespan in the patient's body. They live just long enough to attack the tumor, but they won't persist long-term, meaning you often have to administer repeated treatments.
Wow. So let me push back on this whole manufacturing process for a second. Once we have millions of these cells, whether from cord blood or a cell line, why can't we just freeze them in liquid nitrogen, put them on a plane, and ship them to hospitals worldwide? Like, what is the holdup with just stocking the pharmacy shelves?
It comes down to the microscopic architecture of the NK cell itself. Freezing them causes a very specific type of cryo-injury that T cells just don't experience. You see, NK cells are packed with preformed cytotoxic granules.
The weapons.
Right. Think of them as little internal bombs filled with a toxic protein called granzyme B. When you freeze and then thaw an NK cell, the physical stress forms ice crystals that actually damage the delicate membranes of those internal granules.
No, so the bombs accidentally go off inside the tank.
Precisely. The granzyme B leaks into the cell's own cytoplasm. This causes massive internal damage, and it also leads to fratricide.
Fratricide.
Yeah, where the damaged cells start attacking each other in the bag. When you thaw them at the hospital, a huge percentage of your incredibly expensive therapeutic cells are already dead or dying.
The research outlines a fix for this, right? We aren't just giving up on freezing them.
No. Scientists have engineered a workaround. If you pre-treat the NK cells with specific survival signals, these cytokines called IL-15 and IL-18, before you freeze them, it induces anti-apoptotic genes.
It toughens them up.
Exactly. It essentially transiently reduces how much granzyme B they hold. Then, crucially, after thawing, you let them rest overnight in another cytokine, IL-2, to recover. You also have to absolutely avoid using standard freezing chemicals like DMSO, which actively poisons NK cell function.
So just to quickly recap for you guys listening, we talked about how CAR-T is like that dangerous custom sports car, while CAR-NK is the off-the-shelf armored tank that avoids things like graft-versus-host disease. We've got 4 cell sources: peripheral blood, cord blood, iPSCs, and the NK92 cell line.
Yeah.
And we just covered the massive hurdle of granzyme B leaking during freezing and how we fix it.
That's exactly. So here's a question for you, the listener, to chew on. If you were designing a therapy facility today, would you prioritize the infinite scale of stem cells or the proven algorithmic clinical safety of cord blood? It's a tough call.
It really is.
But let's say we've got the manufacturing down, we've built the tanks, safely frozen them and thawed them at the hospital. Now they have to actually go head-to-head with a solid tumor. And from what I understand, solid tumors are fundamentally different and way harder to kill than blood cancers.
Completely different. Blood cancers are liquid, right? The immune cells just float right through the bloodstream and bump into the cancer cells. But solid tumors actively build a microscopic, highly fortified fortress to keep the immune system out.
Right, and the sources outline 4 major defensive walls this fortress has. The researchers actually use an acronym for it for us to remember, right? VAMP. VAMP, vasculature, acidity, metabolism, and physical barriers. Let's break down exactly how insidious this is, starting with vasculature.
Sure.
How does the tumor use blood vessels to hide? So normally, an NK cell travels through the blood and uses specific surface molecules, specifically ICAM1 and VSNAM1, to anchor onto the blood vessel walls near a tumor.
Like molecular Velcro.
Yes, molecular Velcro is a great way to picture it. But tumors secrete a protein called VEGF. What VEGF does is it circulates to those local blood vessels and completely strips away the Velcro. It downregulates ICAM1 and VSAM1.
So the NK cells flow right past the tumor, unable to stick and migrate into the tissue.
Exactly. They literally can't grab on.
That's incredibly frustrating. Okay, A is for acidity.
Right. Tumors guzzle glucose at an incredible rate to grow, and they excrete massive amounts of lactic acid as waste. This acid bathes the entire microenvironment. When the NK cell tries to enter, it's basically swimming in acid.
Ow.
Yeah. And this disrupts critical internal signaling pathways inside the NK cell, specifically the NFAT and MAPK pathways. It essentially scrambles their internal communications and induces a massive buildup of reactive oxygen species, or ROS, which causes the NK cell to self-destruct.
So they're slipping off the walls and drowning in acid. What about M metabolism and hypoxia?
Because solid tumors grow so fast, they have terrible disorganized blood supply. This leads to severe oxygen deprivation, which is hypoxia. Hypoxia directly reduces the NK cell's ability to produce its weapons, like that granzyme B and another molecule called IFN gamma.
Okay, so their weapons are downgraded.
Right. But here's where the tumor's defense gets truly fascinating. The lack of oxygen also increases the tumor cell's rate of autophagy.
Autophagy, meaning the tumor cell starts eating parts of itself to survive the stress, right?
Exactly. It's regulated by a protein called BCLIN1, but the tumor uses this defensively against the NK cells. If a weakened NK cell actually manages to inject what little granzyme B it has into the tumor cell, the hypoxic tumor cell simply uses autophagy to swallow and degrade the granzyme B from the inside.
Wait, really? It just eats the weapon. Unbelievable.
It neutralizes the bomb before it can go off.
Oh.
And the metabolic warfare doesn't stop there. Tumors also express a membrane enzyme called CD73.
What does that do?
CD73 acts like a vacuum. It sweeps up local ATT energy molecules and converts them into adenosine. Adenosine is highly immunosuppressive. It acts like a powerful sedative. It binds directly to A2A receptors on the NK cell and completely shuts down its function. It puts the immune cell to sleep.
Okay, so the final letter in VMP is P, physical barriers.
Tumors recruit hijacked normal cells, called cancer-associated fibroblasts, or CAFs. These fibroblasts act like construction workers. They remodel the tissue around the tumor into a dense, high-pressure, disorganized wall of stiff collagen.
A literal physical wall.
Yes, a literal wall. And these construction worker cells, along with other corrupted immune cells like Tregs and MDSCs, secrete chemicals. The CAFs secrete TGF-beta, and the Tregs secrete PGE2.
Okay, let's take those one by one. TGF-beta first.
TGF-beta is incredibly toxic to NK cells. It directly inhibits the mTOR pathway, which is how the cell processes energy. It effectively starves the NK cell to death before it can even touch the collagen wall.
Wow. And the PGE2?
PGE2 is another suppressive molecule, though luckily the one can actually be blocked by standard COX inhibitors, like certain anti-inflammatory drugs. But it just adds another layer of defense.
Okay, if the VMP fortress wasn't bad enough, the missing Velcro, the acid, the weapon eating, the collagen walls, I have to ask you about the most terrifying concept in this entire stack of research, trogocytosis. It sounds like something out of a sci-fi horror movie.
It really does. It's the mechanism behind fratricide in the tumor microenvironment.
Explain how trogocytosis turns these engineered super tanks into cannibals, because this blew my mind.
When a CAR NK cell engages in close combat with a tumor, the physical forces are intense. Sometimes, as the NK cell pulls away, it accidentally bites off a piece of the tumor's outer membrane.
Including the specific tumor antigens the CI was designed to hunt, the target itself.
Yes, the NK cell inadvertently wears those tumor antigens on its own surface.
So it accidentally disguises itself as the enemy.
And the other KRNK cells patrolling the area, they see those target antigens. Following their genetic programming, they swarm and slaughter their own teammate.
That is absolutely brutal. So just doing another quick recap for the listener. We have the VMP mnemonic. Tumors cause ICAM1 loss. They pump out lactic acid. They use hypoxia and adenosine to disarm cells. And they build collagen walls with TGF-beta. Plus, we have trogocytosis leading to fratricide.
It's a call.
It really is. So, listener, knowing how actively tumors disarm the immune system, which of these barriers seems like the most critical one to knock out first? It makes you wonder how we even begin to give a therapy a fighting chance against all of that.
Well, that is exactly what the field of genetic engineering is trying to solve right now. Because the tumor microenvironment is so hostile, scientists are genetically hacking the CRNK cells to survive it.
And here is where it gets really interesting, the biological upgrades. How exactly are they hacking them? Let's start with the starvation and exhaustion problem.
Okay, so normally, to keep infused NK cells alive, doctors have to pump the patient full of systemic growth chemicals or cytokines. But flushing the whole body with cytokines causes severe side effects, like capillary leak syndrome.
Where your blood vessels literally start leaking fluid into your tissues. Not good.
Not at all. So researchers are now engineering the CAR NK cells to secrete their own cytokines, specifically IL-15, IL-12, or IL-21.
They basically give the cell a genetic backpack full of its own food supply.
Exactly. The cell feeds itself, allowing it to survive long-term in the host without requiring those toxic systemic infusions.
Brilliant. Okay, next up, they are using CRISPR gene editing to literally delete the breaks from the NK cell's DNA. The sources detail two major targets, genes called CISH and CREM. What happens when we cut those out?
Right, so CISH is a negative regulator of IL-15 signaling, and CREM is a transcription factor that represses effector genes. Essentially, they're genetic regulatory breaks. When an NK cell gets activated and starts fighting, these genes normally step in after a while and say, okay, that's enough, power down before you burn out.
Which is great for a normal immune response so you don't get autoimmune disease, but terrible if you're fighting a massive tumor.
Exactly. By using CRISPR to knock out CISH and CREM, you remove that negative feedback loop. You take the brakes off the cell's metabolism, vastly improving its stamina and lifespan inside the patient.
Okay, so we've given them their own food supply and we've cut the brakes so they don't get exhausted. But how do we protect them from TGF-beta, that energy-starving chemical secreted by the tumor's physical wall we talked about earlier?
You simply blind them to it. By using gene editing to delete the specific receptor that detects TGF-beta, it's called TGFBR2, the NK cell becomes completely immune to the tumor's primary suppressive weapon. The tumor pumps out the toxic signal, and the NK cell just ignores it.
Wow. Okay, but how do we solve the fratricide problem, the cannibalism from trogocytosis? Because you can't just delete a receptor for that.
No, for that, we use something called logic-gated CARs.
I love this concept. It's like a 2-key nuclear launch system on a submarine.
That's a great way to think of it. You engineer the NK cell with 2 distinct sensors. The primary sensor targets the tumor antigen and delivers a kill signal. But you also engineer a second inhibitory sensor that specifically recognizes an NK-specific self-antigen, a friendly marker.
So if the NK cell bites off a piece of the tumor and wears it, a second cell comes up, sees the tumor antigen, and puts the first key in the activating lock.
But before it can fire its weapons, its secondary sensor sees the friendly NK marker on that same cell. That puts the second key in the abort lock. Both must agree to fire. Since the inhibitory signal always overrides the activation, it prevents the cells from killing each other, even if they're wearing enemy camouflage.
Okay, the final upgrade is homing. Because the molecular Velcro is gone, how do we get the cells to actually physically find and enter the tumor?
By engineering specific chemical receptors onto the cell surface. Take glioblastoma, a very deadly brain cancer. It naturally secretes a specific distress chemical called CXCL12. So researchers engineered CR-NK cells to express CXCR4, which is the exact receptor that detects that chemical.
So they act like bloodhounds. They're physically pulled up the chemical gradient directly into the brain tumor.
Exactly. It completely circumvents the need for the molecular Velcro on the blood vessels.
Okay, time for another quick listener recap. We've got self-secreting IL-15 backpacks, CRISPR knockouts of the CISH, CREM, and TGF-BR2 breaks. We have ligated CARs to stop fratricide and CXCR4 receptors for homing. We're basically building these incredible super soldiers.
Yeah.
But honestly, it brings up a huge question for me.
That's that.
We are essentially stripping away all the natural biological breaks of these immune cells. Is there a danger in making a living cell too invincible inside the human body? Like, listener, do you think there's a risk of over-engineering here?
It's the classic Jurassic Park dilemma, right? When you aggressively rewire biology, unpredictable things can happen. And that brings us to what happens when we actually test these engineered cells in human beings.
Right. Moving from the Petri dish to the clinic. What does the landscape actually look like right now?
It is accelerating, but we're still in the early days. According to the data in our sources, there are currently 124 total CAR and NK clinical trials worldwide, but only 39 of those are strictly for solid tumors.
And where are they mostly happening?
The vast majority of this pioneering work is happening in China, and most trials are currently in phase I, meaning they're primarily focused on establishing that this is safe for humans.
The storytelling in these trials is just mind-blowing, though. Let's look at a few specific cases. Let's start with that brain cancer, glioblastoma, and the CAR2 brain trial.
This trial used the NK92 cell line we discussed earlier, targeting the HER2 antigen. And remember, because they used that immortalized cell line, these cells had to be irradiated before infusion. But the delivery method was what made this special.
They didn't just hook the patient up to an IV?
No. During surgery to remove the recurrent glioblastoma, they injected a massive dose of these CAR-NK cells directly into the margins of the brain cavity left behind.
And the results were incredible. 0 dose-limiting toxicities, no inflammatory storms, 0 ICANS neurotoxicity.
And while median survival in this disease is usually very short, one patient actually survived up to 2.6 years, which is practically unheard of in recurrent glioblastoma.
Incredible. And we saw a similar approach in a colorectal cancer case, right?
Yeah, it's a proof of concept case. The patient had a colorectal tumor that had metastasized to the liver. They took an RNA electroporated NKG2D CAR-NK cell.
Okay, hold on. RNA electroporated NKG2D.
Basically, they used electricity to temporarily insert the genetic instructions for a receptor called NKG2D into the cell. They then used ultrasound guidance to percutaneously inject these cells directly into the liver metastasis.
And the timeline of the response here is staggering. Within 4 days, ultrasound showed rapid regression of the mass. At 4 weeks, there was a total metabolic response. When they actually went in and did the pathology, there was 0 viable cancer found in that injected metastasis.
It's a profound demonstration. When you use local delivery, literally physically placing the cells inside the fortress, you can completely bypass many of those tumor microenvironment barriers we talked about. We saw the exact same success in a pancreatic cancer trial, where Robo1 targeting CAR-NKs were injected into a liver metastasis, giving a patient 8 months of survival.
But I have to ask, going back to the Jurassic Park dilemma earlier, Is it always perfectly safe? Is there a cautionary tale in the trials?
There is. There's a rare case in a non-small cell lung cancer trial that proves over-engineering can absolutely be dangerous. The researchers engineered the NK cells with something called a CCCR construct.
How does that work?
They took the external part of a PD-1 receptor, which usually receives a stop power down signal from the tumor, and they genetically fused it to the internal machinery of a 4-1BB receptor, which sends a massive attack signal.
Oh, wow. So when the tumor tries to hit the brakes on the outside, the inside of the engineered cell actually registers it as a command to hit the gas.
Exactly. It weaponized the tumor's own defense mechanism. The problem: it worked entirely too well. The patient experienced a 10-fold spike in IFN gamma and TNF-alpha inflammatory markers.
Meaning
It resulted in severe cytokine release syndrome. The exact life-threatening storm we were trying to avoid by switching away from T cells in the first place. It's the one glaring exception that proves we must be incredibly careful when rewiring these biological circuits.
That is wild. So, recapping the clinical side, we have the GBM CAR2 brain success with local injection, the colorectal liver met complete response, and then the lung cancer CCCR exception proving that over-engineering can bring back CRS. It really makes you think, listener. Seeing the incredible success of injecting these cells directly into the tumor versus relying on an IV drip, how is this going to physically change the way you receive cancer treatment in the future? Like, will the oncology ward look more like a surgical suite?
It's a very likely shift, yeah. Local delivery is proving to be king in solid tumors.
So, stepping back from all the molecular Velcro and genetic hacking, what does this all mean for you, the listener? Let's look at the big picture.
Ultimately, the research points to a clear conclusion. CAR NK cells are proving to be a highly viable, significantly safer, off-the-shelf alternative to CAR-T. Because they don't cause graft-versus-host disease and could be manufactured in massive batches, they have the potential to completely democratize cellular therapy. We're looking at drastically lower financial toxicity and immense scalability.
But I want to leave you with a final provocative thought to mull over. Something the text hints at but doesn't quite resolve. As we move away from bespoke, custom-built therapies like CAR-RT and move toward mass-produced, off-the-shelf models like CAR-NK, we are witnessing the literal industrialization of living medicine. Think about it. If a single umbilical cord blood donation from one mother can be genetically engineered, multiplied, and frozen to cure dozens of complete strangers years later, how is this going to shift our societal understanding of biological donation? How do you regulate a pharmaceutical supply chain when the drug is a living, breathing, heavily armored biological tank?
It's a profound transition. We are truly moving from custom-tailored medicine to universal living drugs.
It's a lot to wrap your head around. We want to thank you for taking this deep dive with us into the microscopic, engineered front lines of the war on cancer. Next time you think of a medical diagnosis, remember that the waters might be murky and the X-ray machine might feel a little broken, but we aren't just looking for a clean line anymore, we're sending in the tanks.